Hi, my name is Jillian Belluck. I am an undergraduate student at Brown University, and this summer I've been spending my time in the Giovannoni lab here at BTI. So first I'll get started with why we're interested in tomatoes and why we're interested in computer vision. So tomatoes have a lot of phenotypic diversity in fruit size, shape, color, leaf size and color. Here you can see an example of cultivated tomatoes, so what you would think of when you buy them in the store, but there are also a lot of wild tomato varieties, and you can see that there's a lot of variation in size, shape, and color. What we're focusing on here is Solanum cheesemanii. It is one of two tomato wild relatives that is native to the Galápagos Islands. These tomatoes are actually a lot smaller than cultivated tomatoes. They're actually the size of blueberries. And what's interesting about Solanum cheesemanii is they don't turn red when they ripen. So some of them stay green, and many of them turn yellow or orange. So we're interested in measuring the variation in these tomatoes. Solanum cheesemanii and the other species native to the Galápagos, Solanum galapagense, are important and interesting because they actually grow in a variety of harsh environments, which makes them resistant to things like salt. So we're studying these tomatoes and we want to see how we can measure the phenotypic diversity. When looking at Solanum cheesemanii, we have 25 different accessions that we're using. This is a map of the Galápagos Islands showing where all of the different accessions came from, and when we're identifying patterns in the phenotypes that we see, we're looking to match them to see if there are any trends from the different islands. And what we're doing with this project, our goal is to be able to phenotype these different tomatoes accurately and easily. So we're using a computer vision pipeline that we developed to take pictures of the fruit and make measurements based on the photos instead of having to do measurements by hand. So this is our pipeline that we're using. First, we harvest the fruit. We tag them when they're at breaker stage, which is the beginning of the ripening process, and then we harvest them 7 days later. Then we take photos of all of the fruit. So we have a photo box here that has a photomacrographic scale. It has rulers that we can use for scaling later on. It has a color checker, and it has a QR code with information about the accession. And we take photos of the fruit. We also take measurements by hand, so we measure the weight as well as the diameter, which we can use for later verification that our computer vision measurements are working properly. And our computer vision tool, the input is an image like this of the whole fruit or of the cut fruit, and we put that into our pipeline to be able to make color and size measurements. So the outputs of our tool are first a proof image. This shows us first that we are correctly finding the color checker and all of the chips in the color card. We use that to color correct the image. We also find the QR code and label that, as well as the rulers in the scale. And then we segment out each fruit individually and label it with the number and put a mask to show that it's found. So this proof image acts as our verification. We also segment out, as I said, the individual fruit and produce photos of those. And then we record measurements of the size, shape, and color of each fruit, as well as save data about the scaling of the photo. And we output matrices that show the average color of each color chip before and after correction, so we can see how our color correction is affecting the photos, and of course the accession information from the QR code. So the first step in our pipeline is to color correct the images. This corrects for the lighting differences that may be in the photos. So this is showing a principal component analysis of each chip in the color checker. So the squares are the correct color, and the circles are showing the color that was measured from the image before color correction. So you can see that there's a lot of variation in the colors. Some of them are not very close to where they're supposed to be, and this shows, for the most part, that the lighting in our photos was too dark. This is a similar plot showing the variation in the color card chips after color correction. So again, these are the same target colors, the squares, and then you can see that for the most part, the color chips after correction are much closer to where they should be. And we color correct not only the chip, the color checker itself, but the entire image to make sure that the colors of our fruit are as accurate as they can be. Next, we're looking at the actual color variation across the different accessions. So this plot is actually showing each individual fruit. Again, we have a principal component analysis, and we took the most common red, green, and blue value in each fruit and plotted that. So you can see that some of the fruit over there never actually turn yellow or orange when they ripen, they stay green, but many of the accessions do turn orange, none of them are red. So this shows us that we are able to identify the colors from the fruit images and plot them to see the variation. And next, we want to verify that our size measurements are actually accurate from the computer vision pipeline. So what we're doing here is we're comparing the measurements that we extracted from the fruit from the photos using computer vision, and we're comparing them to the caliper measurements that we took of the fruit by hand. So you can see this is a linear model showing the correlation, and you can see that our computer vision measurements are as accurate or more accurate than the measurements by hand. We also similarly took the area of the fruit that we extracted from the photos and compare it to the weight that we measured in the lab, and this is a generalized additive model that shows also a very strong correlation. So this proves to us that we can use computer vision as a tool to get accurate measurements on a very large scale with many, many fruit without having to take the measurements by hand. Next, something that we're also interested in doing is looking at the pericarp to diameter ratios of the individual fruit. So the pericarp is this outer flesh here, the locule is the inner juicy part of the fruit, and breeders are interested in the ratio of the pericarp to the locule for many reasons, but from a computer vision standpoint, this is a little bit more tricky to segment out than just the fruit by themselves. So this is what our attempts look like so far, and we're able to get the pericarp from the photo, extract it, and then measure the thickness or the area and compare that to the fruit overall. This is a plot showing by accession the ratio of the pericarp to the fruit diameter as a whole, and you can see that there is a lot of variation across the different accessions. Some of them cluster by island, so for example, the red island, all of those fruit seem to have a larger pericarp to diameter ratio. So this is something that we're looking to move forward with in the future. And next, I just want to show that you can look at the phylogeny of the different accessions. We have some SNP data here that we used to create a tree, and this is plotting some whole fruit as well as some cut fruit across the different islands, and you can see that there are some patterns that we can look for in the size and shape and the color of the fruit. So for example, you see that this clade over here from the purple island all tend to stay green when they ripen, while these clades over here tend to be more orange, especially this island over here. There are also some fruit that stay sort of an orange whitish color, and you can see that the size seems to segment as well between the different clades of the tree. And so just some conclusions that I would like to share is that we found that computer vision measurements of fruit characteristics can be as accurate or more accurate than the hand measurements. And in the future, we're looking to apply our pipeline to characterize a diversity panel of 180 different accessions of both cultivated and wild tomatoes to see the variation in fruit traits as well as leaf traits. And with that, I would like to thank Jim Giovannoni and Vincent Colantonio and Ana Hermans, my mentors, for all of their guidance this summer, as well as the RU program, the Guterman Greenhouse staff, and the Giovannoni Lab. Thank you so much.